So in addition to the work that we've heard from coming out of the Getz lab, there's been a flurry of activity in looking at these different morphotypes over the past couple of years. And Mike Hansen's talked about some of that, and I'm going to touch on using some of his data from that study and a little bit of other data that he didn't present from Great Slave Lake, in addition to the data I'm going to present on Lake Superior. And what caused me to get around to summarizing this talk today was in Andrew's paper, looking at different behaviors and body shapes of these different morphotypes from Isle Royale. Was he mentioned that it was becoming more difficult, it seemed like, to tell leans and Siskowitz apart. And so wanted to look at just one specific aspect of that, and that's lipid content. So we have a nice data set here. We have fish from the 1950s, which I'll explain in a few minutes, data from 1997, and this came out of Chris Harvey's PhD and postdoc project that was the basis for the ecosystem model that Kitchell produced. And then 2013 data came from our surveys, in addition to Wisconsin DNR surveys. Good, pretty much good lake-wide representation for the 2013 data and the 1950s data, and then the 1997 data is all from the Western Isle. So the 1950s data, it was collected from 1953 to 1961, and this was in the Eschmeyer and paper, and they determine these lipids chemically. You see a strong relationship between lipid level and fish length, and that's something to keep in mind throughout this talk when I talk about results, or if I give specific lipid level values. Just remember that there is a strong relationship with length and lipid values, but very distinct difference between Siskowitz and leans from their data from the 1950s. And I'll refer to this data when I say 1955. It's all this data, but it was collected between 53 and 61. For the 1997 and 2013 data, we had stable isotope information from these fish, and then you can convert the C and N ratio from an isotopic analysis to percent lipids. And this has been shown in a lot of studies over time, and it was just recently shown from the EPA Duluth lab by Hoffman et al. To hold true for Lake Superior fish as well. So they looked at values they got from the C and N ratio to those determined chemically, and they saw a less than 1% difference between the two. So lean lake trout, the three time periods, 1955, 1997, and 2013. And again, a strong relationship every year with length, except for 2013. We don't see much of a change in lipid level with length for these lean lake trout. And if we look at from those regression relationships and we calculate the mean percent lipids, so we're controlling for length there for a 300 millimeter fish and a 500 millimeter fish, and just to look at change in average condition over time, very little change. And maybe the striking thing is that 1997 values were higher than 1955 values. And this will contrast with Siskowitz, so that'll make a little bit more sense. And if you can think back when I talked at 11 o'clock and I showed you those plots of crayfish biomass over time, that late 1990s period was one of the highest biomasses that we had for crayfish over that modern day time series from 1970s. So there was a lot of food available at that time. For Siskowitz lake trout, a strong relationship with length for all three time periods. All three time periods were distinctly different, and the trend was with time. Highest lipid level values in the 1950s, moderate in 1997, and then fairly low in 2013. Same thing looking at mean values for set lengths of fish. Not much difference between 1997 and 2013 for small fish, but that difference increases as the fish gets larger. And one of the things that we don't have data on that may be important is we don't have any data on the age of these fish. So if you think back to the plots that that Mike showed of age versus size, and where these fish had grow really slowly after they reach kind of that asymptotic, not the asymptotic length, but that growth really slows down. So if they're like me, right, I've gotten much fatter with age, but I haven't really changed my length. And also think about when, how old these fish were in relationship to the recovery time of lake trout. This will put our data in context with other studies. And again, the thing to keep in mind here is there's two things. One is the change in lipid level with length. So you really need to control for length. And there is a value for the mean length of fish there. But also look, the sample sizes are really small. Basically, there hasn't been much of a change in any of the studies through time for lean lake trout and for Cisco-wet lake trout that seemed like it dropped precipitously from the 1955 sample. Any sample taken like after 1960 is precipitously less than it was before, except for that one sample in 1986. But again, sample sizes is fairly small there. And that was also an unreported length. That's what I was trying to look for. There is, so I don't know what the length of those fish were for that value. So these results made me question how accurate they were. And so another way to look at this is to look at, is there a similar change in body condition? If fat is kind of a surrogate for plumpness of how big a fish is, will we see a similar story when we plot weight versus length? And so this is a plot of weight versus length for our 2013 data, just to go through how I did this. Then if you log that, that's what it looks like. And then you run a regression line through there and you get a nice fit. I mean, it's almost a perfect fit between weight and length. And then from that regression equation, then you can back calculate what those weights were for a set length of fish, so you get a nice smooth line like that. And so not a very big difference for fish less than 600 millimeters in 2013 for the weight to length relationship. And this is kind of the same thing that we saw with the lipids. There wasn't a huge difference anymore between lean lake trout and Cisco-wet lake trout. So to put these data in context, this is the 1950s, so same methods using the length and weight from the 1955 paper and the 2013 paper. So no difference in lean. So we haven't seen a change over time in that body condition index for leans. This is what it is for the Cisco-wet or fat lake trout. So a huge difference, and that matches the difference that we saw with lipids. And then so when I started out, I talked about how distinct are these morphotypes now, and they're not very distinct, right? In terms of when you hold up a fish, the relative plumpness of it, the fat does not seem as fat as it used to be. But leans and Cisco-wets are becoming more similar in body shape. This is the data from the study that Mike Hansen initially mentioned. So these are the fish from Great Slave Lake. So a big difference between leans and fats. And this compares Slave Lake present to 1955 Lake Superior. So leans look like leans, Cisco-wets look like Cisco-wets. The Cisco-wets in Lake Superior are plumper than they are Great Slave, and the leans are a little bit heavier at length than they are Great Slave. So then how do these fish compare to the this is the fish that Mike talked about in his presentation. So this will get at the question, like, how was ours a representative sample of 2013 fish versus the Isle Royale fish, which you would think of as probably a low fished population more than the general population of the lake. And so very similar, exact, basically for leans, and then our fish were a little bit fatter in general, our lake-wide sample, than the Isle Royale fish. And if you look at 1955 compared to the Isle Royale fish, again, it's the same story that we're showing with our fish from when we compare the 1955 fish. So this gives me greater confidence in what we were showing for lipids. We're seeing the exact same thing in terms of the weight-length relationship. So is there any evidence for this, for a decrease in lipids from other lakes or other species? And there's a few that are circled, and those are the only ones that have shown an increase over time. And this data is from NEF in 2012. And these are from a comparison of fish collected from the 1970s through 2008. So every species, almost in every lake, has shown a decline in lipids from the 1970s to present. A summary of the percent lipids, so just so you can get a sense of the magnitude. This is for a 500 millimeter fish based on the regression relationship between lipids and length. And ask a couple questions here. What are the potential consequences now that fats aren't as fat as they used to be? And then I'm going to talk a little bit about what maybe the potential causes are. And those are probably more questions than I'm going to provide answers. So high lipid levels allow the fish, a ciskowet, to swim much easier. It provides them buoyancy, so the energetic demands of swimming are considerably less when you're fat than you are when you don't have that fat. And so this compares calories per hour of swimming to body mass. And if you want to compare for a fish in 1955 to a fish at present at 2013, it went from taking 29 calories to swim per hour to 74 calories now. And another thing to get from this is that a 4 kilogram fish was about 700 millimeters in 1955, and that same fish now is 800 millimeters. So there's just, for the same amount of weight, you just have to be that much longer. They're just much lighter fish than they used to be. So if you're a ciskowet, you're faced with a couple life choices here, that you're not as fat and you're not as buoyant and it's more difficult to swim. You can retain your historic depth distributions and DVM patterns, but increase prey consumption, right? Because you're going to have to eat more because you're burning up more calories. You can retain your depth distribution, but decrease amplitude or frequency of DVM. So instead of going up and down every day, maybe you choose to just go up and down once a week. Or you can move to shallower habitats to migrate shorter distances to get to your prey that may also be going through DVM. So you're going to move from, say, 200 meter deep water out into 100 millimeter deep water, where you can still go to the surface, but you only have to swim 100 meters versus 200 meters. We don't have any data on the first two options, but we do have a little bit of limited data on this third option. And so, this is our trawl survey data, so this is data from 1978 based on those samples all around the lake that the USGS has been sampling, again, since 1978. And this is the percent of hatchery, leans, or ciskowet that are collected in waters less than 80 meters deep. And so we've seen a decline in hatchery fish. They're not stocked anymore, and so they're not around. But we've seen an increase in the number of ciskowet that we've caught over time. And this doesn't seem to be related to water temperature at all. Like, so in that really warm year of 2012, for example, there was more in nearshore waters than there had been in previous years. And you'd think if the water was warmer, they would have moved more offshore. So there's some response here. I don't know what the mechanism is, but there appears to be some measurable response. The fish are moving into these shallow waters. And if you also remember from my talk earlier today, there is actually less prey available in nearshore waters than there is offshore waters. So that adds another kind of head scratcher why they would be doing that. if you eat less, or if you eat lower calorie food, then you're going to lose fat as well. And we have seen a decline in prey, like I said. So those 1997, when lipid levels were high for lean lake trout, that was the highest that we had in our trawl series in terms of prey fish biomass. So that makes sense why those fish were so much heavier during that time period versus today, where there's much less prey available. And also, Cisco, the caloric value of Cisco is so much higher than rainbow smelt. That may play a little bit into it. And we've seen a big decline in the number of corygonids out on the lake. And in some years, we've seen an increase in the number of rainbow smelt available. And the diet studies that have been done in the past has shown, especially for leans, a strong dependence on rainbow smelt. So again, this explains the 97 to 2013 changes, but a little bit, but it doesn't explain the 55 to 2013 big changes that we've seen. There's a few issues with the reduced prey explanations. There's little change in fat or body conditions for lean lake trout less than 700 millimeters. And there's a lack of temporal data for offshore populations, but the data that we've gotten in our recent survey that we've done for 5 years now shows much more prey, 5 times the amount of prey available on offshore as compared to nearshore waters. And this is, you've probably seen this from Patterson's, where they looked at a change in energy density for Lake Superior lake trout, that these were 5-year-old fish, and their explanation for this decline in energy density was prey reduction. So I'm not entirely buying it, but it's out there. We're also seeing a change in environment. I showed this plot as well, where we've seen a change in ice cover on Lake Superior, and this translates to warmer temperatures, particularly in the summer, and they come on, so it's warmer for a longer period of time. And so there's a pretty big difference in water temperatures since 1998. Chuck Medengen and his student Cal from University of Michigan, they modeled the effect of increasing water temperatures and reduced prey for lake trout in Lake Huron and Lake Michigan. And this is, I've redrawn their results from that paper. And so they showed about a 6 to 15% decrease in weight over time, with a 2.4 degree temperature increase and a 10% prey decrease. And this is, and those inputs to the model, that temperature increase is the same temperature increase that we've basically seen on Superior between 98 and 2013. And we've seen more than a 10% decrease in prey availability from the 90s to present. And if you look at, so this is that same weight to length relationship, and for larger fish, we've seen about a 2 to 4% decrease in estimated weight at length for fats, and about a 6% decrease in leans. So it matches these results. They're in the ballpark to their modeled results. The other idea is it could be a loss of genetic diversity in post-recovery populations. So maybe those, the really fat morphotype of the fat morphotype, they died off because of sea lamprey or overfishing. And so what's left, so they're not in the population anymore. And this plot is from the Hansen and Wilberg and Hansen paper, showing the post-recovery populations. And this next plot is from a recent paper that just came out from Bale, and she gave this talk at this meeting last year and showed a decline in genetic diversity for both leans and Ciscoettes over time. And this bottom graph, though, is from Getz, this paper, and where they said, you can correct me if I get this wrong, that homogenization between lean and Ciscoette trout has not occurred. So it's not hybridization is not the reason that we're seeing that similarity between leans and Ciscoettes. So in the end, in my mind, it seems like those lake trout and other people, and like I said, Andrew Muir was the first person that I saw to raise this issue, is that the lake trout in Lake Superior do seem to be losing some of their distinctness in terms of at least body shape, and I think I show here in terms of percent lipids as well. Thank you.